Welcome to Dr. Balaga's God Knowledge Doc podcast, your quick dive into the latest medical breakthroughs to keep you ahead of the curve. Let's get started. This video is on understanding the role of mucins or slime in blood-brain barrier integrity and neuroprotection. The paper was published in Nature recently on 26 February 2025 online. Caroline Bertozzi, Nobel laureate for chemistry, is one of the co-authors of this paper. There was accompanying news and views on this paper by Heidi Ledford. The blood-brain barrier is a specialized structure which controls what enters the brain. It's composed of tightly connected endothelial cells, astrocytes, and pericytes. It protects against harmful substances while allowing nutrients and oxygen. Dysfunction of the blood-brain barrier is linked to neurological diseases like Alzheimer's and Parkinson's. Mucins are large sugar-coated proteins that form a slippery gel-like layer. They coat the blood vessels, creating a glycocalyx that helps to maintain blood-brain barrier integrity, acts as molecular gatekeepers, selectively allowing or blocking substances. Mucins are abundant in young individuals but decline with aging, compromising the blood-brain barrier. Why study mucins in brain aging? Previous research has shown that the blood-brain barrier weakens with age, leading to neuroinflammation. The role of mucins in this process was largely unknown until recent breakthroughs. Mucins may help prevent or slow neurodegenerative diseases like Alzheimer's. If we can restore mucins, we may be able to strengthen the blood-brain barrier and improve cognition. How was the study conducted? They used a variety of techniques, including microscopic imaging, enzyme activity analysis, measured levels of mucin-producing enzymes, gene therapy, blood-brain barrier leakage tests, and memory and learning tests. Evaluated cognitive improvements in aged mice after therapy. Key finding: aging weakens the mucin barrier. Young mice had thick protective mucin barrier. Aged mice showed thin and patchy mucin layer, leading to blood-brain barrier leakiness. Reduction in the mucin-producing enzymes correlated with neuroinflammation. Increased blood-brain barrier permeability allowed toxic molecules into the brain, accelerating cognitive decline. Restoring mucins is a promising strategy. Gene therapy boosted mucin production of aged mice, strengthened the blood-brain barrier, reducing harmful molecular infiltration, lowered brain inflammation, protecting neurons from damage, improved memory and learning performance, reversing cognitive decline. Can mucin-based therapy help humans? Alzheimer's and Parkinson's are linked to blood-brain barrier dysfunction. Restoring mucins could prevent or slow neurodegeneration, might enhance brain drug delivery by selectively modifying the blood-brain barrier. Future research is needed to assess safety and effectiveness in humans. Future research: can mucin restoration be safely applied to human brains? Are there other organs where mucin loss contributes to aging? How do mucin selectively filter molecules? What are the long-term effects of mucin therapy on brain function? There was a recent paper in Nature where in Science Advances where they looked at genetically supported targets and drug repurposing for brain aging from the UK Biobank, and they found 64 genes that influence aging, but they found 13 drugs, including rapamycin, dasatinib, resveratrol, metformin, aspirin, and curcumin, lithium, and telmisartan. And maybe these influence this blood-brain barrier, the mucin barrier. Exercise may also influence it. Key takeaways: mucins plays a critical role in maintaining blood-brain barrier integrity. Aging-related loss of mucins weakens the blood-brain barrier, allowing inflammation and cognitive decline. Restoring mucins via gene therapy improves brain function, showing promise as therapeutic strategy. Further research is needed to explore the human applications and long-term benefits